RNA splicing is an important regulatory mechanism that controls how genes are expressed. The splicing machinery, known as the spliceosome, takes a pre-mRNA and removes from it non-protein coding sequences called introns, and then joins together protein coding sequences called exons. This results in the formation of a mature mRNA, which contains information to synthesize the proteins required for sustaining cellular and bodily functions. Although RNA splicing appears simple, the underlying mechanisms in how this process occurs is quite complex. In addition to the core splicing code and cognate protein partners, primary research has also shown that distinct sequence motifs within exons can enhance or inhibit the inclusion of said exons into an mRNA. These motifs are classified as cis-regulatory elements, known as exonic splicing enhancers or splicing silencers, abbreviated ESE and ESS respectively. The culmination of these various constituents are what leads to the concept of exon definition. As such, the balance between core splicing signals, interacting proteins, and cis-regulatory elements are what establishes the identity of an exon to facilitate its recognition by the spliceosome in RNA splicing. The Stanford lab used a computational approach to identify ESE and ESS sequence motifs that were likely disrupted by disease-causing mutations or by single nucleotide polymorphisms, abbreviated SNPs. These mutations were taken from the Human Gene Mutation Database, and the SNPs were taken from the 1,000 Genomes Project. At the core of this approach, the lab used published datasets containing identified ESE and ESS motifs, one known as RescueSE and the other known as FastHex2. Using a statistical odds ratio to express the likelihood that cis-regulatory elements are affected, the data shown in panel A and B suggests that ESE disruptions and ESS creations are strongly associated with the presence of disease-causing mutations or SNPs. In summary of the publication, however, it was concluded that, as opposed to SNPs, disease-causing mutations were predicted to be most responsible for interrupting cis-regulatory elements. Having introduced the background, my research is centered on taking the work done in my lab and in biochemically testing whether these disease-causing mutations do indeed impact RNA splicing through the disruption of healthy cis-regulatory elements. Using a mini-gene splicing encoder, cell-based splicing assays, and a quantitative fluorescent PCR assay, I've generated a large preliminary data set for the gene F8, in which disease-causing mutations I studied for this gene are associated with the etiology of hemophilia A. In the next slide, I'll present one of several compelling findings to emphasize the impact these mutations have on RNA splicing. So shown here is a data set containing the wild-type sequence for F8 exon 11 and 8 associated disease-causing mutations. To evaluate the statistical significance of these mutants' impact on RNA splicing, I use a conventional measure known as the percent spliced in index, denoted by the Greek symbol psi, which quantifies the percentage of how often an exon is spliced into an mRNA. Among those tested, 3 mutations have the most significant impact on splicing relative to the wild-type control. This control has a quantified psi of 1, indicating that approximately all spliced mRNAs include this exon. On the other hand, the psi values for these mutants were quantified to be less than 1, all with varying degrees of exon inclusion. With all datasets, these quantified results were also visually validated using analytical techniques such as gel electrophoresis to confirm expected mRNA sizes based on our splicing order. In respect to those with a statistically significant impact on RNA splicing, mutants 1 and 2 were predicted to create ESS motifs, and mutant 4 was predicted to disrupt an ESE. These computational predictions support the expectations of what outcomes can arise due to the creation of silencers or disruption of enhancers. To further elucidate the underlying molecular basis and how these mutations impact RNA splicing, the next steps of my work includes understanding the structure of the pre-mRNAs in determining the relevance of said structure in exon definition, and finally, understanding how these mutants affect RNA and protein interactions in the overall scope of RNA splicing. In closing, the broader impacts of this work contributes towards the understanding of how splicing-associated diseases manifest, and then taking those knowledge to potentially learn how to modulate the splicing outcomes to ameliorate disease-associated phenotypes.